De-extinction, the idea of bringing back extinct species, has leaped from the pages of science fiction into the reality of modern laboratories. It's a concept that captivates our imagination, promising a second chance for creatures lost to time. But this isn't about dinosaur theme parks. This is about real science, specifically gene editing technology like CRISPR, which is making the resurrection of species like the woolly mammoth and the dodo bird a tantalizing possibility. Today, we're diving deep into the world of de-extinction. We'll explore the incredible animals that scientists are working to bring back, understand the revolutionary science that makes it possible, and confront the profound ethical questions that arise when we start to rewrite the story of life itself. Are we correcting past mistakes, or are we treading into territory best left untouched? Let's begin with one of the most iconic figures of the Ice Age, the woolly mammoth. These magnificent, shaggy giants roamed the cold tundras of Eurasia and North America until about 4,000 years ago. Their disappearance was likely due to a combination of climate change and human hunting pressure. Now, scientists are closer than ever to seeing them walk the earth again. The process involves taking the DNA of their closest living relative, the Asian elephant, and using CRISPR gene editing to splice in mammoth DNA sequences. These genes code for traits like shaggy hair, small ears, and layers of subcutaneous fat, all adaptations for surviving the cold. The goal isn't just to create a biological curiosity. Proponents argue that reintroducing these megafauna could help restore the Arctic tundra ecosystem. Mammoths were ecosystem engineers, and their grazing and trampling could revert mossy tundra back to grasslands, which might help keep the permafrost frozen and lock away greenhouse gases. It's a bold vision for ecological restoration. Next on the list is the passenger pigeon. It's hard to imagine, but this bird was once the most numerous in North America, with flocks so large they would darken the sky for days. Their population was estimated to be in the billions. Yet, relentless hunting and habitat destruction drove them to extinction in an astonishingly short time. The last known passenger pigeon, a female named Martha, died in the Cincinnati Zoo in 1914. Her death became a powerful symbol of human-caused extinction. The De-extinction project for the passenger pigeon aims to edit the genome of its closest living relative, the band-tailed pigeon. By inserting key passenger pigeon genes, scientists hope to create a bird that is functionally and visually identical to the original. Releasing them back into the wild would be a poignant act of atonement, a way to reverse one of our most shameful ecological blunders. Let's travel to the island of Mauritius, the only home of the famous dodo bird. This flightless bird, or relative of the pigeon, had no natural predators and lived a peaceful existence until humans arrived in the 16th century. Within less than a century, the dodo was gone, hunted to extinction and outcompeted by the invasive species humans brought with them. The dodo became the poster child for extinction, a cautionary tale immortalized in literature and culture. Now, thanks to well-preserved DNA, scientists are working to bring it back. The plan involves using the DNA of the Nicobar pigeon, the dodo's closest living relative, as a template. The revival of the dodo would be a monumental scientific achievement and a powerful symbol of hope for conservation efforts worldwide. From a bird to a big cat, we now turn to the saber-toothed cat, specifically the species Smilodon fatalis. This formidable predator, with its iconic 7-inch canine teeth, roamed the Americas during the Pleistocene epoch. It was a master hunter, preying on large herbivores. It vanished around 10,000 years ago, along with many other megafauna, at the end of the last ice age. While the challenges are immense due to the age of the available DNA, the prospect of resurrecting this apex predator is a driving force for some researchers. The de-extinction process would likely involve using a close living relative, like the African lion, as a surrogate. The ethical debates here are particularly intense. Reintroducing such a powerful predator into a modern ecosystem would require incredibly careful planning to ensure both its survival and the safety of existing wildlife and humans. Our next candidate is the thylacine, also known as the Tasmanian tiger. This was a unique carnivorous marsupial, not a tiger at all, with a striped lower back and a pouch like a kangaroo. It was the apex predator of Tasmania. Hunted to extinction due to a government bounty program that wrongly blamed it for killing sheep, the last known thylacine, named Benjamin, died in captivity in 1936. The tragedy is that the species was declared protected just 59 days before Benjamin's death. Fortunately, a preserved thylacine pup specimen provides an excellent source of DNA. Scientists are using this genetic material to sequence the animal's entire genome. The plan is to use a closely related marsupial, like the Dunart, as a surrogate to bring the Tasmanian tiger back from the brink. Its return could help restore balance to Tasmania's ecosystem. Let's journey to the frigid waters of the North Atlantic to discuss the great auk. This large, flightless seabird was the original penguin of the Northern Hemisphere. It lived in massive colonies on rocky islands and was a powerful swimmer. Tragically, its inability to fly and its lack of fear of humans made it incredibly easy prey. It was hunted relentlessly for its feathers, eggs, and as a food source. The last confirmed pair was killed on the island of Eldey, off the coast of Iceland, in 1844. The de-extinction project for the great auk is considered highly feasible. Its closest living relative is the razorbill, which shares a similar ecology and appearance, albeit much smaller. By editing the razorbill's genome, scientists believe they can create a bird that fills the same ecological niche as the lost great auk. Now for a lesser-known but equally fascinating creature, the auroch. This was a massive species of wild cattle and the ancestor of all domestic cattle today. Standing up to 6 feet tall at the shoulder, these powerful herbivores once roamed across Europe, Asia, and North Africa. The last auroch, a female, died in a Polish forest in 1627 due to disease. While technically extinct, its genetic legacy lives on in modern cattle breeds. This is the basis for the de-extinction effort. Instead of gene editing, this project uses a process called backbreeding. Scientists are selectively breeding rustic, hardy cattle breeds that retain strong auroch-like traits. The goal is to create a new breed, the toros, that is physically, behaviorally, and genetically as close to the original auroch as possible. These animals could then be used in rewilding projects to restore natural grazing patterns in Europe. Next, we have the quagga, a subspecies of the plains zebra, distinguished by its unique striping pattern. Unlike other zebras, the quagga was only striped on the front part of its body, with the stripes fading to a plain brownish color on its hindquarters. It was native to South Africa. Hunted to extinction for its hide, the last wild quagga was shot in the 1870s, and the last individual in captivity died in an Amsterdam zoo in 1883. At the time, people didn't even realize it was a distinct subspecies until it was too late. Similar to the auroch project, the quagga project uses selective breeding. Since the quagga was a subspecies of the plains zebra, its unique genetic information still exists within the broader zebra population. By selectively breeding zebras that show quagga-like characteristics, the project has already produced foals with the distinctive faded stripe pattern, effectively bringing the quagga's appearance back to life. Let's head to Spain to find the Pyrenean ibex, a subspecies of the Spanish ibex. This mountain goat, known for its large, curved horns, was native to the Pyrenees mountains that straddle France and Spain. Its population dwindled due to intense hunting pressure over centuries. In a landmark but ultimately tragic event, the very last Pyrenean ibex, a female named Celia, was killed by a falling tree in 2000. However, scientists had taken tissue samples from Celia before her death. In 2003, they made history by successfully cloning her, implanting a nucleus from her cells into a goat egg and using a domestic goat as a surrogate. A female Pyrenean ibex was born, marking the first time an extinct animal had ever been brought back. Sadly, the clone had a severe lung defect and died just minutes after birth. Despite the failure, it was a monumental proof of concept that de-extinction was, in fact, possible. Finally, we consider the moa. This group of 9 species of giant, flightless birds was native to New Zealand. The largest among them, the South Island giant moa, could reach up to 12 feet in height and weigh over 500 pounds. They were the dominant herbivores in New Zealand's ecosystems for millions of years. But when the Maori people arrived, the moa, having never faced a terrestrial predator, were hunted to extinction within 200 years. The prospect of reviving the moa is challenging but not impossible. Extraordinarily well-preserved bones, feathers, and even dried muscle tissue have yielded high-quality DNA. While finding a suitable living surrogate for such a unique bird is a major hurdle, some scientists speculate that a large, flightless bird like the emu or ostrich could be a possibility after significant genetic modification. The return of the moa would be a spectacular revival of a lost world. The science of de-extinction is undeniably thrilling, holding the promise of righting historical wrongs and restoring lost biodiversity. But it forces us to confront some of the most profound ethical questions of our time. Is it right to spend millions on bringing back a few select species when thousands more are currently on the brink of extinction due to lack of funding for conventional conservation? Should we be focusing our resources on protecting what we still have? Furthermore, even if we can resurrect an animal, can we truly resurrect its species? A species is more than its DNA. It's its behavior, its social structures, its learned knowledge passed down through generations. An animal born in a lab will not have a mother to teach it how to find food, avoid predators, or migrate. And what about the ecosystems we would reintroduce them to? The world has changed. Habitats have shrunk, and new diseases and predators exist. Are we bringing these animals back only for them to suffer and die out again? Or worse, could they become invasive species themselves, disrupting the delicate balance of modern ecosystems? This is the great debate of de-extinction. It's a powerful tool, but one that carries immense responsibility. We are on the verge of being able to play God, but the question remains, should we? The conversation is just beginning, and its outcome will shape the future of life on our planet. If you found this exploration into the world of de-extinction fascinating, make sure to subscribe to Infinitunial for more deep dives into the wonders and complexities of our universe. Thanks for watching.